This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 585, recorded on January 31st, 2020. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be back with functioning tech this time.
Very good. Good luck.
Yeah, thank
Keep your
you.
fingers crossed.
Exactly. And it is on the okay side here. It's 6 degrees Celsius, a little gray out.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's a nice 34 Fahrenheit, 1 degree Celsius, and partly sunny.
Are you sure it's not partly cloudy?
No, it's partly sunny. I'm optimistic. It really does. You can see shadows. So, yeah.
We have 5 degrees today and totally cloudy. No sun at all. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 58 degrees Fahrenheit. That's 14 C. That's chilly for us, but probably fine for everybody else. A few puffy clouds. It's a nice day.
And from Western Massachusetts, Alan Dove.
Good to be here. And we've got a wind out of the south about 5 knots, visibility 10 miles, overcast at 6,000 feet, and temperatures 4 C, dew points -3.
I was over on Reddit the other day. I posted last week's podcast, and I noticed there were some comments, and one person said, it's a good podcast, but they're a little chatty.
What are you supposed to be on a podcast?
No, exactly. Are we supposed to talk or not? Do you want silence? We can give you a nice hissing noise.
That's right.
I have some of that.
For an hour and a half. Anyway, we had an amazing podcast last week, and I just want to say that's like the biggest podcast we've probably ever made in terms of people listening to it. Got a lot of downloads. So there's nothing like an outbreak.
Yeah.
I mean, among the top listened to episodes are mostly outbreak episodes. So I wanted to say two things before we ask Kathy about ASV. Today is actually the deadline for the abstracts, right?
Correct.
Today being Friday, so when people hear this, it will be too late. I have a note here that I meant to read. So when I was in Sweden, the second podcast I did, a graduate student, Sindhu Vangetti, sat in, and she told me she's a longtime listener. And then a few weeks later, she sent a note. Dear Vincent and the TWiV gang, I'm about to defend my PhD thesis thanks to TWiV. Hope you enjoy reading my thesis. Thanks for your inspiration. The weather in Stockholm right now is 1 degree C and sunny. And she sent a copy of her thesis. So as in other places, the Karolinska, they make a little book out of it. So she sent me this. It's called Monocytes and Dendritic Cells, Roles During Human Influenza and Hantavirus Infections. So thank you, Sindhu. She has written in before. And her PI was on that episode, actually.
Ah,
Cool.
Congratulations to all.
Yeah.
Yes. And now this is already a few months old, and I've been neglecting to read it. So there you go. Okay, Kathy, what's new with ASV?
What's new? Well, we are accepting late-breaking abstracts on the 2019 novel coronavirus. Submission will open February 10th and be open until May 1st. So that could be for oral or poster presentation, but the only ones accepted at that late date will be on that topic. And we haven't really talked about some of the features of the meeting. Anne Palmenberg is going to be the keynote speaker, so I think that's pretty exciting. And we have 5 satellites, and
That's
those are
solid GPS fix.
Yeah, right. They're on a variety of interesting topics: shaping the infected cell proteome, frontiers in invertebrate virology, co-transmission of multiple virus genomes, viral evolution of immune evasion, and viral modulation of host cell metabolism. So those are on Saturday before the main part of the meeting. And if you sign up for one, you can float among them. You could mix and match if you want, or you could just sit in one entire satellite for the whole morning and early afternoon. So maybe that's enough features for this week. We have to save them for next time.
Okay. Well, we have a whole lot of coronavirus information to give you today. I want to start just by saying, so I've had quite a few media contacts this week, and I was asked by Fox News to appear yesterday on some live noon show called Deep Dive, and they were going to have 3 ex-scientists, experts, and the moderator was going to be from the Wall Street Journal, and I said no, because I don't believe Fox News is a bona fide news channel, and I don't want to have any part of it. I know I should have gone and been the real news, but I actually didn't trust them to do anything appropriate. So
yeah, that's a tough call.
It was. I thought about it very hard, and pretty much everyone I spoke with agreed. And I wanted to tell everyone that I think they're part of the problem with our country right now, and I can't be part of that. I mean, I will talk to most outlets. I talked to Time this week. I talked to Newsweek, a radio station in Boston. I talked to a Chinese news outlet. So I'm happy to do it, but, they all seem bona fide, and so I don't know what happened, but thank you for the invitation.
I've gotten a couple of inquiries on Twitter asking me to appear as some kind of virology expert, and I point them to you and Ralph Barrick and Matt Freeman.
Yeah.
Because this is not, I'll provide commentary, but I'm not going on the radio as some kind of coronavirologist.
So what would you do if Fox called you? Point them to someone else.
I'd say impeach that. No. I would point them to somebody else, definitely.
Yeah, so I just wanted everyone to know
this was a product of me having said something on Twitter that sort of went viral, and then I got
So I just wanted you to know that I will work to speak for virology, but in the right places, which may not be perfect. But the other thing I wanted to point out is, as we are reading a lot about coronaviruses, everyone seems to capitalize the C. And here on TWiV, as you know, we are sticklers for virology terminology, and our advisor is Jens Kuhn, who I've learned all my taxonomy, capitalization, italics, and I've learned from Jens, like from genus up, species up, actually. You have italics and capitals. So, like the Coronavirus family would be capitalized and italicized. The genus beta coronavirus, of which the new virus, SARS, MERS, etc., they can all be capitalized and italicized. But when you start doing the vernacular, like coronaviruses, it's not capitalized. There's no need to. And this 2019-new coronavirus is just a temporary name. It's, it's not a species, even so you don't have to capitalize it.
And at least one of the early papers that kind of said we're calling it this used novel instead of new.
Right. It's not novel. Yeah.
Right.
It's a good thing, because every time I use novel in the textbook, Jane Flint yells at me. Can't use novel. I use it a
textbook, not a novel.
Okay. A couple of interesting tidbits here. Now, someone, maybe it was Michael Schmidt, pointed me to this wonderful Johns Hopkins website. It's called 2019 nCoV Global Cases. And there you have a map, and you have confirmed cases by country and region. You have deaths. You have recovered. You have a graph of the cumulative cases. And right now it's reading 9,776 cases confirmed and 213 deaths. It's quite a cool site.
It is. Because I'm always looking around to get the latest. And it gives you a date line on that. That's as of January 30th today. Oh, no, wait a minute. January 30th, is that yesterday?
Yes.
at 9:30 p.m. Eastern Time.
Right.
And each of the little panels you can expand and fill your screen with it if you'd like.
And you can click on the red dots and find out where they're referring to.
Yeah, so you can go around the world and click and see how many. So it's quite good. I would recommend it highly because it seems to be that they put up the cases as soon as they happen, right?
Mm-hmm.
So anyway, check that out.
What we haven't been able to find is the daily caseload.
Yeah.
Right?
Yeah, traditional
a graph.
A traditional epi curve where you have now in some of the papers there are such curves for just a small amount of time early on, but they don't have it for every day. So I would really be interested in that. At some point, I suppose we'll see one, but don't have one right now. There's sit reps, of course. Someone wrote in and said, "I want to hear a sit rep from Rich every TWiV now."
Well, the sit reps are quite long and detailed.
Yeah.
Yes.
They are, and they of course the numbers are not as good as that Johns Hopkins because they're a little behind. So the sit rep came out yesterday. It only has 7,800 cases. Wow, this is really increasing quickly, right? Mm-hmm. But they often have some good information in these sit reps. Oh, here's an epidemic curve, but they're outside of China. Yeah, these sit reps for some reason have...
Yeah, weird. Yeah, you get the daily caseload outside of China, but not in China.
Yeah.
I don't get it.
Well, maybe there are too many. I don't know.
I mean, you could. I've thought about this. You could make your own. It's the first derivative of the cumulative curve, right? But
I don't know what you said there.
But
I mean, you could make your own. But
it's the rate of the first derivative is the rate of change. So the rate of change in the cumulative would be the daily rate. Yeah.
Oh, you can do that from the Johns Hopkins data because I think if you hover over each point, the number comes up. Okay.
If you go on the explainer page, they add new stuff all the time, and now they have new cases day by day. And so, Rich, you might like this graphic. It's got
wait a
red
minute. The explainer page. Where are you?
The South China Morning Post explainer page.
That's really good. Okay.
Yeah, and scroll down, and now they've got. It starts out with a few dots, and then they fill in and mainland China versus the rest of the world. Yeah.
Oh, so they have the daily epi, right?
They don't have a graph.
No.
Yeah, so the one above is still cumulative, where it compares it to coronavirus, right?
Yeah.
Oh, that's a good one because you can see corona spiked and went down very quickly, and this thing is boom, goodbye.
Yeah.
It's the peak is way past corona already, and
you mean past SARS?
SARS.
Yeah, past SARS. Yeah.
SARS is the light red one. Cumulative.
Yep.
Yeah.
Yeah, this is a cool site.
All right, so before we have a couple of papers. Gosh, the papers are coming out like gangbusters now. Hard to keep up. But before we do that, I just wanted to point out, I went over to the CDC site because I was curious how much flu there was this year. There have been 15 million cases of influenza, 140,000 hospitalizations, and 8,200 deaths.
Just in the USA.
Yep.
Right.
Yep.
Yes.
That's a lot.
Yeah.
Yes.
Yes.
You think they're going to quarantine travel in the U.S.?
Because of flu?
Just kidding. Of course, it's everywhere, so it wouldn't help. But
right.
And also as we'll see later, the fatality rate for flu is less than. Well, I don't know what the current coronavirus is. It looks like about 3%, but it's kind of early to do that. But it's
we'll get into that later, I think.
Flu is less severe. But the point is that if this new corona should stay with us and become endemic in humans, which is possible, and it causes a lot of cases every year, people will forget about it, right? It'll be just another viral illness that happens regularly. This is news because it's unknown. We don't know where it's going, right?
Well, and even saying that it'll happen regularly might be a bit of a stretch because people who've had this presumably have developed immunity, right?
Yeah, that's the other thing. We're going to at some point.
So this is not, as far as I know, coronaviruses don't shift the way flu does. So we're not going to get a new strain of this every single year.
But Alan, does a virus shift in the woods?
Yes.
So I'm thinking here about Zika.
Mm-hmm.
Okay. Did that wasn't ultimately global, was it? It went from Africa
Pretty much.
through the South Pacific, and then
as long as the mosquito or the travelers were there, yeah.
Okay, and then South America,
went all the way around. And now it's burned out.
Burned out partly because of what Alan was talking about,
immunity,
which is immunity.
Yeah,
everybody who was going to get it has been exposed and either got over it or didn't, and now there will be some level of new cases of people getting exposed.
Presumably now it's some version of endemic, and non-immune people will come along and be exposed to it, but we won't have this big blast.
Yeah, I think that it'll. And we don't know what percent of the population has to be immune to stop transmission. It varies. This is typical for enteroviruses too. There are outbreaks every couple of years because you have to build up a non-immune population for them to infect. But we don't know what that is for this virus. But I presume it'll eventually get there. And I just wonder also, it's currently winter in China, and I wonder if this is going to be a winter illness. It's going to be seasonal. But so there are some people making projections of 100 million infections. They just take the doubling time and they keep going forever. But it's not going to go forever. At some point,
epidemiology is not quite that simple.
No, there are a lot of factors. So it will stop at some point. And SARS stopped very quickly. But there are other reasons for that, as we'll talk about. So just after we finished last week, a whole bunch of papers came out. And I just wanted to focus just on a couple of them because they're quite interesting. I think the listeners would like them. The first is a virology paper, which is on bioRxiv. I assume it's going to come out somewhere else at some point. It's called Discovery of a Novel Coronavirus Associated with the Recent Pneumonia Outbreak in Humans and Its Potential Bat Origin. So this comes from quite a few investigators from the Wuhan Institute of Virology and Chinese Academy of Sciences in Beijing.
And I was pleased last week to hear Ralph talk about the Wuhan Academy and the scientists there and say that these guys are right on the money.
Yes.
So it's funny, the Newsweek...
first author on this is Peng Zhao. Senior author is Zhengli Shi, and there are gazillion other authors.
There's two first authors, too. Xinglao
Yang is also.
So Newsweek was particularly interested in this lab. They said, anything about this Wuhan virology? Are they good? Do they know what they're doing? Do you think they drop viruses? It's just people just want to know screw-ups, don't they?
Well, this one was the one that David Quammen referred to. And she, Zhengli Shi, is the woman who was on the 2005 group showing the SARS spillover from bats, and then was the one working in 2017, collecting bat feces, published in 2017 after 5 years in the Yunnan cave, the multiple coronaviruses in the 4 bat species. And one of the ones that's 96% identical to this 2019 coronavirus.
Right.
So she is somebody to know.
Yeah.
The paper.
Yeah, so to be clear, there's a virology institute in Wuhan because it's a good place for a virology institute. That's not the reason these viruses are breaking out there.
Not at all.
Oh.
No. This is clearly not a lab virus. They didn't even have this virus. She had, as we'll see, she had to get the whole sequence. So this paper is fascinating because it's not very old yet. It says in the beginning, there's no large-scale human-to-human transmissions so far. Things change so rapidly. But they took...
when was this?
Posted January 23rd, but they would have been writing it leading up to that.
Probably the night before,
Yeah.
So they took samples from 7 patients, and 6 of them were seafood market peddlers or deliverers. And they have a bronchoalveolar wash. They put some fluid way down into the lung and wash around a little. And then extract it, and they did some PCR, and they said we're a coronavirus lab, so we used coronavirus PCR primers, and they found positivity, and then eventually they got the entire genome out, 29,891 base pairs, and from that you can see it's clearly a SARS-like coronavirus, and it's very similar to one. It's the most identity, 96% to one bat isolate that was found in 2013 in Yunnan.
Right, in that cave, right.
And they have...
What I really liked is that she said, we found a hit from the polymerase in our database.
Mm-hmm.
So they just had from what I understood, they didn't have the whole sequence of this bat virus.
No.
Okay. They had a
right.
partial sequence that was the polymerase.
Exactly.
And they got just a taste of that from this, and then went back and sequenced the whole thing. And the similarity is striking.
Mm-hmm.
Yes.
So obviously they had this, whatever sample they had from bats, they had it frozen, and they said, let's go back, and they got the whole genome from that, which is really remarkable, right?
Mm-hmm.
Mm-hmm.
So yes, these two viruses, this bat virus and the new coronavirus, they have a common ancestor. Okay, so this relationship doesn't mean that the new SARS coronavirus came from the bat virus. They have a common ancestor. That's all you can say because they're so related. And then they
are so closely related, though. It's I think pretty safe to say that this new coronavirus came from a bat virus.
Oh, for sure.
Oh, absolutely.
But not this one that they have.
This specific. It is not this specific virus moved into humans, but it's something very close to this.
Yeah, they have what we say is they have a common ancestor.
Yes.
Mm-hmm. And yes. So this is one reason why it's not a snake virus, because
yes,
snake coronas are different. It's not a lab virus because they didn't even have this virus in the lab.
Right.
It also is much closer to, excuse me, Brianne, much closer to this bat virus, if you look at this similarity graph they have, than it is to SARS that went around. So this is, it's a new virus in humans. Is that fair to say? Yeah.
Mm-hmm. Yeah. Sure.
I think so. This morning in my virology class, we discussed a different one of the recent papers that came out about this virus. That was in the New England Journal of Medicine, and they also had a really nice phylogenetic tree showing how closely related this virus is to a lot of bat viruses. Mm-hmm. And my students were very, very struck by it, and they were very angry that people were saying that it was a snake virus after looking at that figure.
There you go. Good. I mean, these bad viruses have been known for some time. The EcoHealth Alliance has been funding work to capture bats and sample them, and they published a paper in PLOS Pathogens in 2017, a rich pool of bat SARS-related coronaviruses, and I think one of the authors are here on this paper as well.
Yes,
and one virus was pulled out from this study, crawling into caves and sampling. So
this is why we do the basic science.
Yeah,
you have to do it. Yeah, and it's also why you need to. Before you say things, you have to read the literature, right? You can't just say things like, oh, there are snakes in the market. Maybe it's a snake virus. Well, anyway. This paper does a couple of other interesting things. They do some, so they isolate the virus. And this is a lovely experiment. In my virology class, we had just talked about using cell culture and CPE, cytopathic effect, to detect the possible presence of a virus. And so they took this lung wash and they put it onto Vero cells, vervet monkey kidney cells, right? Vervet.
Vervet.
I told my class that. I didn't say African green. I said vervet.
Vervet.
And HUH cells, these are human liver cells. And in 24 hours, you can see cytopathic effects. Perfectly classical cell rounding and detachment. They have a picture of the cells in there. And I showed it to the class. And I said, you see, I'm teaching you stuff that people actually use. Not just
that must have been a good day in the lab.
Can you imagine how excited they were to see this?
Oh, yeah.
to put this on a couple of cell lines, see if you see CPE, and of course, whoever's doing that was saying, yeah, right, whatever, I'll go ahead and do it. And then you come in the next morning, whoa, it worked.
They also had some antibodies against the conserved protein of a coronavirus, and they can stain the infected cells. You can see nice immunofluorescence. They did some electron microscopy. You can see typical coronavirus morphology. They didn't.
The thing I was going to say, because you tweeted about having the CPE as a nice classroom thing, in the figure where they use a serum from one infected patient and look at it over time, and they assay the IgM and the IgG. It's the perfect textbook curve of what that should look like. That's in figure 2B.
Yeah, exactly.
Love it.
They also do neutralization assays where they take serum from patients and they mix it with this virus, and it neutralizes infectivity as you would predict it. Oh, by the way, they took the supernatant from those cells. They sequenced the whole genome.
Mm-hmm.
Say, the virus that's the virus.
Yeah.
Which is just great. Oh, and then finally, this is beautiful. They have, the SARS virus receptor is a protein called ACE2, angiotensin converting enzyme 2. They have HeLa cells that would have it or do not have ACE2. And they have ACE2 from humans, Chinese horseshoe bats, civets, pigs, and mice. And they show that if ACE2 is present from all but mouse, this virus will get into cells.
So let's talk for a minute about the significance of ACE2, because this keeps coming up, and frankly, to tell you the honest truth, I'm a little vague on it. Is this potentially a boundary to spillover? That is, do all of these bat viruses use the ACE2 receptor? Is there some adjustment required, mutation drift from bats or other species to infect humans that involves the receptor?
So it can be a boundary, right? It can prevent crossover. Not all of the viruses circulating in bats bind to ACE2, especially human ACE2. Right?
Okay.
That's what we're looking for here. So the fact that this one does
is significant.
And this was suggested by some other work that we mentioned last time, but here they're actually using the infectious virus, which is the ultimate experiment. I think that I think some of the other ones were great, but this is the proof. In fact, Ralph Barrick published a paper last week, bioinformatic analysis, suggesting that ACE2 is the receptor. So this is beautiful.
Yeah.
In fact, the paper we talked about of Ralph's a couple of years ago, they showed that some of these bat SARS-like viruses can infect human cells, and they infect via ACE2. And so that, not all of them, but that's a concern, of course, because that allows entry at least. Whether the virus whether the human cells are permissive or not is another question, but getting in is a step, right?
And do we know in this case, whether the bat virus that is so similar to this novel coronavirus has sufficient similarity so that we can say that the bat virus could use the human ACE2 receptor?
So in that paper, people have looked at that. I don't know if this particular virus, it's been done, because remember, they didn't even have the whole genome. They just had a piece of the polymerase, right? So they certainly don't have virus, and they didn't have the whole genome until they did this here. So I'm not sure we know it for that particular, but we do know that, I would guess if it's 96% identical, it's likely that it could bind ACE2, but you just need one amino acid to make it not bind. So I don't know the answer to that.
And importantly, as you point out, the binding and entry are not necessarily the only barriers to a spillover from an animal to a human.
Right.
So the experiment to do would be to isolate that original bat virus and try to infect human cells or other cells that the novel 2019 virus gets into, and see if the infection goes to full course.
Right. Stay tuned. I'm sure we'll see.
Yes, I'm sure that is underway.
So before we move on, I want to tell you about why they picked up this this cluster of pneumonia cases. There's a New England Journal article, which another aspect we're going to talk about. It's called The Early Transmission Dynamics in Wuhan, China of Novel Coronavirus-Infected Pneumonia, which, I'm sorry, the title is just not working for me. The virus cannot infect pneumonia, right? So anyway, it's just me. I know, I'm picky.
It's fine.
No, that's a good point.
In fact, there was a early paper from the Chinese CDC. Let me give it to you here because it's quite interesting. Just give me a moment while I find my paper, my coronavirus 2019. No, that's colony collapse. That's not coronavirus. Chinese CDC. Here we go. And this Chinese CDC Weekly, the novel corona genome, and they say the disease has been subsequently named novel coronavirus infected pneumonia. I don't think that's right. No, that's not right. So fix it. Anyway, this paper says why they picked this up. So the earliest cases were identified through the, quote, pneumonia of unknown etiology surveillance mechanism. This is an illness without a causative pathogen that fulfills certain criteria, radiographic and temperature, so forth. And this was put together after SARS to try and pick up any new kind of pneumonia that arose. And so then if you get a case that fulfills these criteria, and that's what happened here, then they put together a field team. They have people from China CDC with provincial, local, municipal CDCs and prefecture CDCs, and then they coordinate field investigations. They collect specimens, and those are sent to Beijing for testing. And so that's what happened here. They had this surveillance, which is really good, right?
Yeah, that's
unusual pneumonia. If we can't figure out what it is, we're going to look for SARS-like viruses. So I thought that was cool.
Yeah.
In case you're wondering why. And so the Newsweek person said to me, so these people in the lab who worked on this, were they just not doing anything and then they gave them this to do? I said, no, they were working in their coronavirus lab. They're working, and then they were told, you have to investigate this. That's how it works.
Yeah.
You're a reporter. Aren't you working on one story and then another story comes along?
Were you doing nothing until this story came along?
No, it's so funny. The way you put it was like, did they have nothing to do? So obviously many people do not know how science works still, right?
Right.
Yeah.
Okay, the other paper that I think is...
Wait a minute.
Yes.
I'm actually, I hadn't looked at the New England Journal paper, but I'm just looking at it now. There's a feature of Figure 1 that's really interesting to me. First of all, there's your date.
Let me see if... Yeah, go ahead. Yeah, Figure 1 is cool. Go ahead.
So first of all, there's your daily case rate, except it looks... on the graph, after it peaks on January 8th and decreases, but they have a note here that says that after January 8th, that's likely to be due to delays in diagnosis and laboratory confirmation. But what interests me is that they've split these out into linked to the Hunan market and not.
Yeah.
And if you look at the December cases, there's a fair percentage that are linked to the market. And then when you get into January, boom, there's still a trickle of those, but there's an explosion of cases that are not linked to the market.
Yes.
So that's like, it's a good visual of, regardless of where it might have originated, you see the human transmission taking hold.
Yeah. Yeah. So this one is actually not fully correct, and that's why I wanted to talk about one of the Lancet papers that came out last Friday. As we were talking, Ralph said a Lancet paper came out this morning, and I didn't have time to read it. One of them is Clinical Features of Patients Infected with 2019 Novel Coronavirus in Wuhan, China. And they have, so these are, how many patients? 41 admitted patients on January 2nd. And they studied them, and they have an epi curve, okay? And they have a similar thing. They have a bar graph of people with or without Huanan seafood market exposure. And there are two things we learned from this that are really cool. First, the first case was actually December 1st. Now, if you remember, they were saying 20th or 21st originally. And obviously in testing, they're finding earlier cases. So this is December 1st, and it has no connection with the seafood market.
Yeah. Yeah. So what happened here is the cases that were picked up by this pneumonia surveillance program that we just talked about were the mid to late December cases, and a bunch of those people were linked to the Huanan market, and those were the ones initially, okay, it came from the market, yada, yada, yada. Not to be confused with yada, yada, yada virus, by the way. So now they're going back and saying, all right, let's trace this back. Let's look at older blood samples that were taken for other reasons or what have you, and see what the earliest case is that we can find. And that's how we get back to December 1st.
And so that means this person was probably infected in November.
Yes.
And some other analyses that have been done, they now have multiple virus isolates and multiple sequences, and they can backdate to the possible most recent, and it could be as early as October. And so this virus, so what's emerging is that this virus may have been circulating earlier than we think. Maybe there were cases of pneumonia that weren't picked up. And then that's why there are more people actually infected, and that's why it exploded all of a sudden in January.
And that implies, certainly this graph implies, that it didn't come from the Huanan seafood market.
Did not come, right. In fact.
So it got, probably what happened is it came from somewhere else, and then somebody at the seafood market had it, passed it to other people. Right. And so then, it kind of came from the market in the sense that, the flu that you caught came from the school, but that's not where it originated.
Yeah, so that first patient, December 1, had no connection with any of the other 40 patients in this study.
Right.
And about 13 of those had no connection with the market. And so this was covered in a Science article last week, and they had a quote from one of the Chinese researchers, and he said, we really don't know where this started.
Right.
One thing that is sort of interesting is that there's been an analysis that's been around on Twitter that showed that they've done some environmental sampling at the market. And they've seen that a good proportion of their samples do come up with this virus. And so it's sort of interesting to think about, was there a super spreader at the market, or was there something that amplified a little bit at the market? But
what kind of environmental samples do you know?
They don't really say. It's very unclear what's going on. They say environmental samples from the market, and 33 of 585 samples were found to contain nucleic acid of the coronavirus.
What's the source of this?
I linked it in the Google Doc.
It's the new Twitter Journal of Science.
Right.
There are 2 or 3 different reports, all from Chinese media sources. And they say that all of those were in the western zone of the market, nearby one another.
Well, there are obviously other markets in Wuhan, so I'm sure they're looking like crazy to find out where this. We'll find out eventually, I suppose. So that was an interesting revelation from this paper. The other is that if you read into the discussion, they start talking about antivirals. No antiviral for corona has been proven to be effective, but there's a drug which Ralph mentioned last time, remdesivir, which is a nucleotide analog, which has been shown to be effective for MERS and SARS coronavirus infections. In this paper, they say A randomized controlled trial has been initiated to assess the efficacy and safety of a different drug, lopinavir and ritonavir, in patients. So those have been tested in the hospital where these patients were. Lopinavir is a HIV protease inhibitor that seems to have broader inhibitory activities, and ritonavir is a very interesting drug. It is originally devised as an HIV protease inhibitor, but it turns out it inhibits a cytochrome P450 that inactivates drugs. So if you give it along with any other drug, it makes them more potent. Anyway, it's being tested right now in China, so that's good. And hopefully they'll test remdesivir also, because this could be a quick solution for very serious patients, right?
Yeah.
I think that's very promising there.
So now another thing that comes out of these analyses and showing that it's been going on longer than we thought, and you've got a lot of circulation happening here, is that the size of this outbreak is certainly bigger than what we see. So we're looking at the 9,000 or whatever figure that we quoted earlier. Those are people who have tested positive for this virus. And in order for that to happen, they have to have come to medical attention, and somebody has to have decided to do this test. There's a huge population, I don't know how big, of people out there who didn't come to medical attention and therefore didn't get the test, right?
Yep, exactly.
Yeah, the extent of that is unknown, but that's, I'm sure they absolutely do exist. How many could vary hugely, right?
So when we get into talking about things like the case fatality rate, that's when we say case fatality rate, that means people who came to the hospital and then died versus people who came to the hospital and didn't die, or who were tested for this virus.
Yeah.
It does not mean, if it comes out to 2%, it does not mean 2% of the people who get this virus die. It just means of the ones we detected who had this virus and who were sick, that's the percentage that dies.
It's the same issue with avian influenza, H5N1, right?
All of these, yeah.
Has a apparently high case fatality, but those are the people who are sick and go to the hospital. And then maybe a lot of people who get infected, not that sick, and they don't, the denominator doesn't account for them. Hello, Dickson.
Hello there, everybody.
Hi, Dickson.
Do you need some technical assistance?
I always do. I have to get my pacemaker working.
Well, please do. By the way, if you need a pacemaker, you're one of the people who would have a problem with this new virus.
Yeah, I've read the Lancet report that just came out of the first 48 patients, and it was very interesting to see all the symptoms that they exhibited, including diarrhea.
Two main ones are cough and fever.
That's right.
That's
right.
And then I think muscle aches. And
myalgia.
And one other, which is escaping me, but it's okay.
Was it dyspnea?
I don't know.
Yes. You're absolutely right.
Shortness of breath.
Shortness of breath, right.
Exactly. And forgetting which seal was the biggest.
Right.
That's a very common symptom.
It is.
So the reference there is that the leopard seal is not the biggest seal.
That's right.
Is it nasty nonetheless?
Oh, what?
The elephant?
The leopard seal is nasty.
Leopard seal is nasty. The elephant seal is pretty docile, I think.
Except for the cute one that my brother sent us the picture of. That
a cute leopard seal?
Yeah, it was pretty cute.
How about that?
Looked mellow.
So let's talk a little bit about reproductive number and fatality. Because we have now a preliminary reproduction rate from this paper that Rich was talking about, early transmission dynamics. They were able to, so early in the outbreak, before you really know what's going on and you impose any quarantine or measures of any sort, you can get a idea of the intrinsic reproductive capacity of the virus. In other words, how many people will one infected person infect, right? And so in our discussions on the Google Doc, we have come to learn that. So R0 is the basic reproduction rate, which is in the absence of any intervention.
Right.
Correct, Dr. Condit?
Yep. That was my understanding from reading all this stuff that's been posted on the Google Doc.
So we have a Google Doc that we all use to prepare a show, and someone will post something and someone will correct it, and we go back and forth, and it's really a learning experience. It's wonderful. And then we leave the paper trail in there so you can see.
Exactly.
Someday to publish all these, right?
That's right. That's right.
There are a lot of them. 500 and what?
85.
85?
Yeah, or more.
And they go back to number 1, because I don't know why, but I started doing it on day 1.
That was a smart thing to do.
So their number, this is the New England Journal, Early Transmission Dynamics, 425 cases. They get about 2.2.
Fatality rate?
No, reproductive number.
I'm sorry.
Reproductive
Sorry.
Reproductive
So to be
number.
clear, that means that on average, every infected person, on average, would infect 2.2 other people.
Yeah.
Okay.
And then
average is important because different viruses under different conditions will behave differently. You know, we've talked about super spreaders. You know, SARS, there were some people who infected a lot of people and others who didn't infect any. Okay, so it's an average.
There's a range, of course, for that. And the Times today had a nice graph on its front page where they graph this reproductive number versus, I guess, case fatality. But in terms of reproduction rate, you know, it's sort of in the same ballpark as SARS, right? Mm-hmm. Even seasonal flu is a little lower, it looks.
I think this is an enormously informative graph.
MERS just seems to be a little lower, less than 1, which is why I guess you don't. So if you go at 1 or less, then you don't sustain an outbreak, right?
Right.
Yes.
Correct.
Right.
So MERS is less than 1, and these chains of infection are very short.
Stop kissing your camel.
And importantly, okay, the R naught is as we have already said, the basic or intrinsic reproductive rate. If you then intervene with public health measures and quarantine and that kind of stuff, you can reduce the effective reproductive rate. And the idea, I guess, would be to get it down to less than 1.
That's right.
To squash the outbreak.
Yeah. So the reproductive rate is basically dependent on, how long an infected person has time to contact other people, right?
That's right. Mm-hmm.
They're shedding. If you have a week, it's not good. If you have a day, it's good. So they're working really hard to identify cases and put them away, stay home, right?
Exactly.
I think that this graph is a really nice image, and I would suggest that everybody take a look at it. I have had to explain that whether or not something is contagious and whether or not something is deadly, those are not always related to many of my friends who are not scientists. And so to see this so prominently in the New York Times explaining that exact fact made me really happy.
Can we use that as a show image without any sort of copyright?
Probably not without stepping on some copyright.
Okay. By the way, they must have modified this from this link that I found.
Yes.
I think so.
Yes.
Okay. Which is a more expansive version of the same thing, which is just as interesting. That's amazing. What a cool graphic. I notice on that graphic that I found, if you look at the ordinate, it is nonlinear. It's not even exponential. It's just plain old all over the place. They spread the numbers out in whatever fashion was convenient. But it works okay.
I want to point out that on this reproductive index graph, the winner is measles.
Measles.
Like 15.
Yeah.
Yeah. But on Rich's, malaria and rotavirus are even a little bit higher.
Yep.
Oh.
The other thing on this graph is the case fatality ratio, right? The number of deaths divided by the number of infections, and usually that denominator is the cases that you detect and diagnose, right?
Right.
And the winner here seems to be bird influenza and Ebola viruses, MERS, smallpox. They're very high. They look like 50-ish% or more, right?
Yeah.
And then they have polio and Spanish flu at 10%, SARS as well.
Mm-hmm.
And then this new corona is below. There's a big range, right? Because we don't really know yet, as Alan said. We don't have the numbers yet. And it goes from, it looks about 7 or 8% down to 0.1%, depending on what numbers you're looking at.
Mm-hmm.
And seasonal influenza is about 0.1%. Common cold is 0. Nobody dies of the common cold. You do?
No, but you feel like it.
Right.
No, common cold isn't so bad.
Yeah, I
Is
got
it?
the sniffles.
Measles is about 0.2, it looks like. Anyway, it's a really good graph. So we don't know this number yet for this new corona, but we'll know at some point.
If you just take the, just for a frame of reference, if you take the numbers from this Johns Hopkins site that you cited earlier,
Yeah.
and just divide the total deaths from the total confirmed, you come out with 2% currently, okay? With all of the caveats that we've talked about, okay? That is, this being the diagnosed cases, et cetera. So I would say that's a maximum-ish at this point.
I don't know why they got this big range. I guess they're looking at different studies, right?
Mm-hmm.
Different populations. Okay. Now, someone put in a lovely graph here. It's titled, Quarantine of Movement Would Have Limited Effects.
Oh, I did. And this is from the thing that's called Nowcasting Forecasting. It's the link that's right above it that you had put into the show notes.
Right.
From
yes, that's right. I remember that being in there. Yeah. So they did some predictions of the effect of quarantine on the extent of the outbreak. And if they didn't quarantine Wuhan, it looks pretty similar to quarantine in this graph, doesn't it?
Yeah, and they go out to August. So, yeah, this is a presentation by Professors Gabriel Leung and Joseph Wu at a media conference January 27th at the University of Hong Kong, and it's a WHO collaborating center. So it was some kind of PowerPoint. I don't know how you found it, Vincent, but there it is.
And this is based on years of research on the effectiveness of quarantines and modeling of this, and people have looked seriously at whether this type of thing can even work. And the answer is basically no.
Yeah.
It came from ProMed Mail. A lot of rich resources over there. There's just a lot of it. And I made the mistake of having them send daily email digests. And it's just a lot of
I get those as well.
And I know now why I stopped years ago, because it just
fills up your mailbox.
But for now, it's good. Yeah, this is really good. A lot of people who have talked to me have asked, are these quarantines effective? And last time Alan discussed this and talked with Barrick, and it's not clear that they have an effect, especially if you announce it the day before and 4 million people leave the city, right?
Now, where the hell did they go, by the way? 4 million people in one day?
That's one of the many problems with trying to do these travel restrictions and such.
Good
is
Lord.
that you cannot keep track.
No.
So if you had kept everything running and said, okay, we're going to screen everybody at train stations and airports and look for people with fevers, that might have some kind of an effect. But if you say, okay, we're going to shut down the transportation system and nobody's allowed out, everybody flees and you can no longer track any of them.
Correct.
And if you get enough people out, then that's it. The virus is everywhere. Right?
Well, of course it is.
And that's what we have now. It's in many, many countries.
Did you see Tony Fauci last night on the NPR News?
No.
I did. And he said exactly that. He said, we can't manage China. He says, what we can do is we can manage the cases in other countries, particularly Western countries, that have good medical facilities, and you can treat palliatively and cure a lot of these people.
So Delta and American just shut down all flights to China.
And I had a meeting in April canceled in Nottingham, England, of all places. By April, people will forget that it was corona.
This may be not so virulent, but it's going to have a big economic effect.
Billions and billions and billions.
So those two airlines shut down travel to China in general?
I don't know how long, but here's the news thing. Well, it's gotten blocked by other stuff now. So Where is it? View. I don't know how long they plan to do it. It just the alert I got from the Times said they shut it down.
Okay.
It can't be forever.
Yeah, Delta, American, and United are suspending all China mainland flights.
Wow.
Until March 27th.
That's incredible.
Well, by then it'll be over, right?
Well, maybe.
Well, that's just, I mean, if it's not, then maybe they extend it. But they're saying that for now, for their scheduling purposes, they need to pick an end point.
That's a big deal. I have to know.
It's a very big deal.
It says here that this comes after pilots with American Airlines filed a lawsuit demanding the airline stop flights to the country.
Oh, my gosh.
Right. So they're dealing with their flight attendants and airline and pilots unions who don't want to get sick, and they certainly don't want to fly into a city that suddenly gets locked down while they're there, which is a risk, right? You know, if you were flying into Wuhan and you did your layover and you're ready to catch your flight out the next day, and then all of a sudden they shut down the airport.
Exactly.
Yeah, Delta is going to run through April 30th.
That's incredible.
But
that is a massive economic hit for the airlines and for China and for the airports where those flights originate. And
that's right.
Yeah,
I know that, and we've talked about this. Apple has the 30 seats on every daily flight from San Francisco to Shanghai booked because they had people go, and that's all. It's all gone.
And by the way, I tweeted about this because I covered it a couple of years ago for an article that I was doing on antibody validation. When you order monoclonal antibodies for your lab, you open up a catalog and you say, "Oh, I need monoclonals or I need antibodies anyway against this or that antigen." And a lot of things, commonly studied antigens, I'm sure ACE2, which we were just talking about, you can order antibodies for it. That's all great. You can order them from a bunch of different vendors. What most people don't realize is that the vast majority of those are produced by biotech companies in Wuhan.
Really?
Yeah.
And are there? It is to biotech what Shenzhen is to electronics.
Wow.
So you have these white label manufacturers who actually produce the antibodies, and those are then relabeled by all of your major lab suppliers or many of your major lab suppliers and sold under different brands.
Sure.
The reason I was writing about it at the time was because people will often say, "Oh, well, this antibody from that vendor didn't work. We're going to try, getting an antibody."
We're all the same from the same place.
And they don't realize they just bought the same darn thing and spent possibly thousands of dollars buying duplicates of the thing.
Oh, brother.
All that stuff made in Wuhan. I don't know what the status of it is going to be as the city is now locked down.
Mm-hmm.
So there could be knock-on effects in your lab. When you try to order antibodies, they may be out of stock. I don't know.
So is China stopping outgoing flights?
I don't know.
The Dow Jones is on. Go ahead.
Yeah, so I know they've stopped regularly scheduled outgoing flights from Wuhan Airport, which means I don't know what effect that has on cargo. If they've also stopped cargo flights from going out, if they're quarantining air crews in that regard, and certainly as the city remains locked down, things are going to happen that people won't be able to show up to work and make the antibodies.
That's right.
Yep, it's a problem.
The Dow Jones is off 550 points.
What?
Nearly 2%.
Dickson, there goes your retirement.
Yeah.
It went a long time ago.
Oh my gosh!
So it's that
this is billions.
I mean, it's
and
sort
billions.
of weird, sort of justice to have a virus trash your retirement.
Yeah, especially for virologists.
Yeah.
Hey, Rich, you and me both.
Oh well. Guess we'll just have to keep working. Well, I will, because you guys are already retired.
No. I'm still at Fordham.
I want you to double my salary.
Yeah, that's right. That's right.
Okay. Let's up the donations here to TWiV.
That's right.
A couple of articles.
I got some time before my retirement.
Yeah, you do.
Well, Brianne, your generation, my generation, we're not going to be able to retire because the older generation will have looted all that. So
yeah,
really?
no, I wasn't planning on it.
Yeah.
Kathy, you put a Yang article here. You want to comment on that?
Oh, it's just another article about R naught, and I thought it was well put together. He doesn't make the distinction toward the end of R naught and R0, or R naught and R.
Yeah.
But basically, he says that R naught can be altered by public health measures, and it's just really nicely spelled out for the lay reader, I think.
So David Quammen had an op-ed in the Times the other day where he said, this is a problem. We have a big world with a lot of mouths to feed, and we have to change our ways. So I wrote and I said, Ralph said this is a luxury meat that they're eating at these markets, right? It's not to satisfy a protein need. The real problem in China is all the pigs that they raise, and
they're gone too.
So he wrote back and he said, I'm between planes in Atlanta, and I don't actually remember what I wrote. I wasn't talking just about China. In places like Congo, Amazon, parts of Borneo, West Africa, wild meat is indeed an important protein source. That's probably what keeps Ebola going. And
Western Pennsylvania, upstate New York.
Upstate New York, it's a big thing.
You got it.
Yeah.
Really?
And in Tennessee,
wild protein?
No,
deer?
It's all throughout the South. Also, come on, wild boar hunting is a big deal.
Yeah,
Okay.
Now I was just picking some northeastern states because people don't usually think of that.
Yeah, that's all true.
Then we have a hand washing article from Kathy. Should we wash hands, Kathy?
Yeah. Well, first I want to go back to David Quammen's because he talks about, the fact that we could move quickly on some of these things because of dedicated scientists and outbreak response medical people at Wuhan Institute of Virology, EcoHealth, U.S. CDC, Chinese CDC, and so forth. But he talks about how funding this is really important. And so the story I heard on NPR this morning is how the funding for U.S. preparedness is set to run out in May. But maybe now that won't happen because
everything else is running out in May also, by the way, I hate to tell you.
Yeah.
Well, not too long ago, we used to have a program called Predict to isolate viruses from different sources and sequence them. And that was canned. The administration took away the funding, short-sighted stuff. Because when you go in caves and look for viruses, that's how we're ready to work on this one, right?
Right.
Yeah.
Yep.
So this article says washing is...
so this is an article by a physician who's now a journalist with the New York Times. And she talks about when she was living in China during the SARS outbreak. And the international school where her children were students was one of the few that stayed open through the outbreak. And some of the simple precautionary policies, they sent a note to parents not to send a child to school who was sick. And the students were screened at the door with ear thermometers. There was no sharing of food at lunch. And the teacher led the kids in frequent hand washing throughout the day at classroom sinks while singing a prolonged hand washing song so that they didn't just pass their hands under the water. And if a family left Beijing and came back, the child would have to stay at home for an extended period before returning to class. And the thing that's really cool is she said, with those precautions in place, I observed something of a public health miracle. Not only did no child get SARS, but it seemed no student was sick with anything at all for months on end. No stomach bugs, no common colds. Attendance was more or less perfect.
Woof.
Yeah.
And
Semmelweis was right.
And so, one of the important things about this article is she's saying that they didn't flee the country. They didn't really very substantially change their lives. They just took some common sense precautions.
Right, and they didn't wear masks. So this made me wonder what effect this would have on influenza to have these kind of control measures going on for this coronavirus. Thinking about, is this going to change the epidemiology of that? Because of, if people did institute hand washing and public health measures.
Yeah. Yeah, because people don't take the flu seriously enough to do it, but maybe coronavirus will scare them into actually washing their hands.
But I don't know why that would prevent all transmission, because as we know, it's respiratory aerosols, right? But obviously there's an important component
keeping sick kids home is also a big deal.
That's a big deal. Yeah. And I also think kids touching their mucous membranes and then touching each other probably is a big deal too, right? And adults, of course, do not do that. So
No.
Never.
Well, I tell my students that if you're out in public and touch the subway pole or whatever, just don't touch your eyes or nose until you wash your hands, right? That's common sense.
Easy to say, hard to do. Today in my class, I had one woman who admitted that she was sick and enough with influenza last year to go to the hospital.
Okay.
So she's in her late teens, early 20s, and looked in good health, but obviously got very sick.
Well,
Sorry.
You need to stop this.
Thank you.
Hey, you feeling okay?
You feeling any myalgia?
Stop it. Stop it.
He's coughing. Got fever.
No, I told you about last week, didn't I?
I'm going to put a thermal scanner in my office. So
you do that.
Come in here. I want to. There's a nice quote in Vincent Munster wrote a commentary in New England Journal to accompany one of these articles. And nice quote here, which summarizes nicely this balance between virulence and transmission. He says, if this virus transmits efficiently, which it seems to be doing, its seemingly lower pathogenicity as compared with SARS, possibly combined with super spreader events, could allow large-scale spread. In this manner, a virus that poses a low health threat on the individual level can pose a high risk on the population level with potential to cause disruptions of global public health systems and economic losses. That's exactly what we've been saying, right? Disruption. But it's a good point that low virulence is okay. You can still spread viruses quite effectively.
Yep.
In fact, better.
For class.
Now, this the New England Journal paper that we've kind of referred to a few times, besides calculating the R0, they also calculated the incubation period, the time before symptom onset, right? And the mean is 5.2 days.
Right.
With an interval of 4 to 7 days. So that the question there, of course, is whether you are transmitting the virus during those days of incubation. And it varies according to virus. And last week, remember, Ralph Barrick said for SARS, the most transmission is at the peak of disease.
Huh.
And he said those people are very sick. They tend to be hospitalized, and that's why most of the transmission
hospital workers.
is hospital. And here we're seeing with this, it's really community.
Yes.
So people are not that sick, and they're walking around in transmission. But there was no hard evidence for transmission from asymptomatic individuals. And just this morning, a paper came out in the New England Journal. It's apparently rushed out very quickly. And it is a transmission of the virus from an asymptomatic contact in Germany.
Yes.
And this 33-year-old healthy
This is really cool. Figure 1 is really good.
They have this is like one of those correspondences, from a bunch of physicians. Mm-hmm. So
by the way, I think all the papers we're talking about on this episode are open access. So if anybody clicks those links, normally New England Journal has a subscription requirement, but all this stuff they're doing the right thing and putting online for free. Sorry, back to the paper.
No worries. This is about a business person who got sick on January 24th. He got a fever and a cough, and then he recovered and he went back to work. But before he got sick, he had attended meetings with a Chinese business partner, and a resident of Shanghai had visited Germany earlier, and she was there between January 19th and the 22nd. So remember, he got sick on the 24th. So this is before he got sick. She flew back to China, and she got sick and tested positive for the new coronavirus.
What I really like about this is that when she was in Germany and had contact with this person, she was asymptomatic and didn't get symptoms until she flew back to China. So there's no question about whether this person contacted her when she had symptoms.
So she could have been the first person to introduce this virus into Shanghai. How ironic. Via Germany.
Right.
Anyway, so then they went back, and he turned out to have the virus as well. So that's the first evidence of asymptomatic transmission. So it's happening. This is not going to be an isolated case, right?
I think every possibility is there, right?
But Tony Fauci makes a big point that the asymptomatic cases are not going to be what drive an epidemic.
Yeah, that's true. I would still
Well, they do for polio. because 1 in 100 get paralyzed. Everybody else.
Yeah, but you get it from the environment, not from shaking hands.
Yeah, in this case, it's correct. It's not going to drive it, but they will make silent transmission, right?
Yes.
And that's why if someone
Look
goes...
at AIDS. AIDS is even worse. HIV/AIDS. Come on. 8 years of incubation period.
Now, we got quite a few emails after last week's. So let's see if we can get through a few of them.
Before we leave the general thing, I just want to ask, I don't really have an answer to this, but historically, how frequently might something like this have happened that we just didn't notice in the same way, didn't register? I mean, some of this has to do with the fact that we've got surveillance now. We have the techniques for diagnosing what's going on, for identifying the virus. We have a lot of communication, et cetera. I don't think, my guess is that this stuff has been going on all the time.
Mm-hmm. Yeah.
Right?
Yeah. And we have, I mean, simultaneous with all the scientific ability to discover this, we have developed the technology for this worldwide transportation network that can put anybody anywhere in 24 hours.
Mm-hmm. Exactly.
And
that's
massive population density.
Yes, and we've had a vast increase in the global population. And so I think altogether, that's what's driving these global pandemic fears and legitimately so.
Another thing that Ralph mentioned was the density of the bats crowded together, and different species of bats with lots of different viruses. And so if you encroach on their environment, which we are doing as humans, then that could possibly increase the number of those kinds of events that happen. But I agree with you, Rich. I'm sure that this kind of thing happened in the past. We just
this has been going on
couldn't
forever.
See it.
Right.
Oh, I mean,
and something like Ebola or Zika virus, those I'm sure have been spilling over into humans for a very, very long time, and just finally, caught a flight.
I mean, look.
So all of these things, the factors that we're talking about, population density, travel, et cetera, can work to aggravate the situation. But it's not as if we have all of a sudden entered a new phase of human existence where this sort of thing is happening. Okay, certainly these situations are aggravated, but they're not entirely novel. Okay,
right,
going on.
No. So we had smallpox many years ago, right?
Well, the best example I know of that has nothing to do with what we're talking about right now is when the Spanish first came to the New World.
Yeah,
there were 50 million people here. When they left, there were 5 million people.
Yeah, the viruses the Spanish brought,
they didn't
never
kill
seen
them
before. Yeah, because you couldn't get across the ocean.
The other thing is that they hybridized, if you wanted to look at it this way, or cross-married, or whatever they did, in order to take their genes and leave them here. And so now you'll never get that to happen here again. So that was.
So these things have always been happening, but they weren't noticed, and they weren't as extensive, less population, right? You can look in history, you can see polio way back to.
Or epidemic typhus.
Yeah. So it's just now that, but the question of whether they are more frequent now is a good question, but hard to answer because we don't have historical data. Right?
We don't have the past info.
Yeah. But I don't know what the solution is. I think as long as you harvest wild animals, you're going to put yourself under the threat of infections. I don't know what you do with it.
That's an easy thing to point to, but even if you stop that, we're still building out into areas where the bats live, and we're still going to have contact with bats, and it's not just bats, and it's
look what happened in Australia.
Well, Nipah and Hendra,
we have a lot of contact with a lot of animals now, and
Nipah and Hendra, we didn't eat the bats. They came to eat the mango trees.
We didn't encroach on the bats in Australia this last fire season. That was something different, and that also alters the distribution of animals.
Well, that's going to change their habitat. They may go onto farms now to find food.
Exactly right.
And that's what happened with Nipah. They were burning forests, and the bats were displaced. They went out to the pig farms, right? So yes, you don't just have to eat meat. I agree, but that's a component, and
sure,
it depends on the mechanism of transmission, right?
I think all we can do is surveillance and public health measures.
Yeah, wash your hands.
Exactly.
Yes,
One thing I know about is that the rate of trichinosis went down in China as a result of swine flu because they lost their animals.
Okay. All right. Let's read a couple of emails. The first one is I found a website where someone listened to the whole episode last week and kind of summarized it, very detailed.
Yeah.
He wrote, with information flying rapidly around, it was time to get some facts from the virus experts at TWiV, who interviewed Ralph Barrick. And then he summarized it, or she. It's great. It's a really good job.
Nice.
Just a weird website. The ads are kind of weird, but
It's a great summary.
Great summary. Thank you very much, whoever you are. We appreciate it. All right, Norman writes, great to hear you guys talking about microbiology and current concerns in such a relaxed, frank, and informative discussion. That's all that most lay people want to hear, I believe. Just open, honest facts without hyperbole or evasion.
Mm-hmm.
This is something so lacking in today's media that people are forced to Google for true expert perspectives on virtually everything of importance. So many thanks to you and your guest for the day, Ralph Barrick. One comment, having once lived in China for about 7 years, I'm sure you are right about the cultural nature of these live food markets and the need to mitigate rather than ban this age-old practice. Hopefully the younger generations in China and elsewhere will be more receptive to the educational health warnings about the dangers involved and the need to change from potentially harmful habits. Programs of education should also be on the menu. Norman is in South Wales, UK. I have to say that a lot of my students from China have no idea about open meat markets. They say they go to the supermarket. Yeah. So I think a generation is going to change this probably. Yeah.
I was listening to NPR, and they replayed a program where they talked about a book by Deborah Blum, and it was because there was going to be a feature on PBS this week about food safety at the turn of the 1800s to 1900s.
Yes. The poison eaters. That's right.
Yes.
The poison squad.
The poison squad, exactly.
I read it.
Yes. One chemist's single-minded crusade for food safety at the turn of the 20th century, and how food safety did change. You know, there used to be stuff, milk was watered down with pond water and other things like
worse
that.
Than that.
Yeah.
It was really
chemicals too.
Pond water was watered down with milk.
Right. So anyway, it made me think, well, maybe this can change. And like Vincent just said, with a new generation, people maybe will see that this is something that's important.
Wasn't Sinclair Lewis the author of that book, The Jungle, that had something to do with the meat market?
Upton Sinclair.
Upton Sinclair. I keep blocking on those names.
Yeah. I mean, that's changed a bit, but it's still pretty gruesome, right?
It's very gruesome.
Dickson, can you take the next? No?
I don't have it up on my screen. I can't get connected.
You forgot your password, huh?
I did.
Okay. Alan, can you take the next one?
Sure. Seneca writes, hello, as someone who deals with images, I can say that according to the images submitter, the image is an illustration of Mycobacterium tuberculosis, not a virus. And this is the image we were complaining about where they were presenting this thing as... Yeah,
it looked like cheese sticks.
Right. It looks like cheese sticks, and it was presented as an image of a virus. And if you reverse search on TinEye, you can get the original Shutterstock photo, which is tagged bacteria, Mycobacterium tuberculosis, causative agent of tuberculosis, 3D illustration.
I didn't know.
Who knew? TinEye, man.
Yeah, that's cool. I didn't know that.
Really cool.
All one word. Yeah, it
Nice.
looks like it's free site. It's pretty cool.
Cool.
Yeah.
Kathy.
Yeah, photographers often use that to find people who've pirated their work online.
Yeah, okay. Stacy writes, hi all. I have a few questions about the coronavirus episode. 1, are bats getting sicker around the world? 2, are some bats super spreaders? 3, are fruit bats the usual hosts for viruses that spill over? 4, can TWiV invite a bat specialist? I am addicted to your show. I do extremely tedious art projects. The only fun is in the planning. And listening to your conversations feeds my brain while my hands are occupied. Thank you.
Cool.
Yeah.
You should get Peter Daszak on here.
Done.
Yeah, Peter. So, are they getting sicker? My understanding is not from viruses. I mean, they're okay with these viruses, and that's one of the questions we have. How do bats live with all these exotic viruses?
Exactly.
And Lin-Fa Wang is one person, and there are many others studying that.
And that's TWiV 296.
So cool.
And I interviewed him in Australia a while ago, but I think it's still a relevant bat episode because he is a bat specialist. So I would check that out.
Bats are in fact getting very sick around the world, and there's a fungal disease called white nose syndrome that's wiping out entire hibernacula, and it's widespread, certainly around the U.S., which is a huge ecological problem. But that's not to do with things like the coronavirus spillovers.
No.
I don't know if bats are super spreaders.
Probably there are some.
I haven't heard.
I mean,
there's
probably some variation from one individual bat to the next in how much they spread a particular virus.
I mean, Dickson, do you know the range of bats? Some are small and some are long range, right?
They're huge. Oh, yeah, that's exactly right.
I mean, some just
about
stay
30
in
feet with a good arm.
Some of them stay. Some of them stay in their cave, and others go around. Right.
Some of them travel over 50 miles every day.
There are migratory bats too, right?
Yeah.
Well, a lot of them do migrate. And I saw a program, I won't tell you when and where, but it was late at night when I wasn't sleeping very well one night. And I watched a program on a University of Pennsylvania, not the University of Pennsylvania, but a university in Pennsylvania, a woman who specialized in the white nose disease. And it's traceable back to Europe.
You watch too much TV.
No, but there's no suffering from it in Europe. It's just
right.
part of their environment.
Okay.
But when they brought the bats over to the United States, somebody went to a bat cave, she thinks, in Europe, and then came to a bat cave in the United States and brought it, and that started the whole thing. In Albany, of all places.
Are fruit bats the usual hosts for viruses that spill over? No, they can be. So Nipah and Hendra came from fruit bats. A lot of paramyxos, but these in China are horseshoe bats.
Right.
Right.
They have lots of teeth, so they
Two
must
different
eat
kinds.
meat.
And there's a difference.
What do horseshoe bats eat? Are they fishers?
I don't know what they eat.
Or do they eat moths? They're big. I mean, they must have a...
I think they're insectivores, aren't they?
I don't know.
That's a good question.
20% of mammals are bats, according to Peter Daszak.
Yes, of course. Vincent Munster wrote something about a bat. Let me see if what he wrote me is interesting.
Yeah. Wikipedia says horseshoe bats eat insects.
Okay.
From stones and branches. That's the lesser horseshoe bat.
Yes, Chinese horseshoe bats are insectivorous, highly active. And coronavirus versus fruit bats, putative phyllo reservoir, less active. So some bats. Okay. Anyway,
the fruit bats do migrate because the fruit is not always on the same trees at the same time.
They go get the fruit. That's right.
They do.
You can't do one, Dickson. Rich, can you do the next one?
Yeah, this is
Chase.
Chase writes, so I just started listening to your podcast with the coronavirus, and I was wondering if there is a difference between the effectiveness of these two face masks in the case of an influenza outbreak. I have been taught by some that surgical masks are good to use if you are infected, but not that efficient in preventing infection compared to an N95 or N99 mask. Is there anything conclusive on that? Okay, so we talked a lot about these during the 2009 influenza and epidemic.
I found an article that says they're both similar. The question is it better than nothing? And I would guess it is because they're going to stop the respiratory droplets, right?
Yeah.
Right.
So Michael Schmidt said his hospital in North Carolina can't get any surgical masks. They're all sold out everywhere. Can you imagine?
And several articles that I've read about this talks about the sort of casual layperson's use of these things and highlights that it kind of depends on the circumstances and how you use them. They have to fit right. You have to, in the appropriate circumstances, have them on all the time. You can't, scratch your nose under the mask. So
pick it for that matter.
The point is that a lot of people don't use the masks appropriately. Okay, if they're used appropriately, they can be a help in situations where you're likely to have exposure.
We should all just wear spacesuits and we'll be fine.
I've thought for a long time that we all ought to wear helmets all the time.
Yes.
And I'll sell them to you.
The difference in the numbers has to do with that the 95 filters out 0.3 micron particles, and the N99 could filter out 99% of particulate matter. But so you might think that would be better, but that's so much denser that it gets sweatier and more moisture laden, and so to know which one's better.
As somebody who occasionally wears one of these for doing stuff around the house or woodworking or something, they're not real comfortable. So if you were doing something really dusty, I have some N95 masks and just with my safety goggles and such, and I'll put one on. But if I'm out mowing the lawn or something, gosh, it's hot and really unpleasant. And so you pay a price for the N99 masks. Being even denser, you're going to have even less air coming through. It's going to be even less comfortable.
Yeah. Wash your hands.
Wash your hands.
Exactly.
Yes.
On the other hand, you can get some pretty cool face surgical masks with things on them.
Yes.
Very nice. Brianne, you're next.
All right. Sophia writes, "Dear TWiV team and dudes, Vincent started this. Sorry. Thank you for a very informative episode 584 on coronaviruses. You asked most of the questions I would have asked, and your guest was wonderful, having enjoyed him on previous episodes as well. So I feel a lot better about this epidemic now. Thank you so much. Okay, so now I would like to ask some of the questions of the general public nature, I guess, if you'd like to answer. 1. If you do get sick with the flu during the year, months after you got vaccinated, you have mild symptoms. Are you still infectious? And for how long? I think I am sick with the flu now and not sure if I should show up for work in a school-type environment."
So I actually asked this of Kramer. Not Franz. Florian. And he said, if you have symptoms, you're likely shedding.
Yes.
And so you should stay home.
Stay home.
Absolutely. 2, if SARS ended up causing 800 deaths worldwide, but flu kills around 300,000 each year, why make such a big deal about the coronaviruses and not the flu? There was a lady guest on BBC that actually said that the general direction to the public would be to worry more about their yearly flu shot than NCOV.
Absolutely.
Yes, I agree.
The host
point.
The host last night asked Tony Fauci that very question. And he said, Tony agreed. He says, you fear the thing that you don't know, and you're not afraid of the thing you do know. So you know a lot about influenza, so you expect it, but you don't know anything about corona.
My students this morning asked that question too.
But if this really is 2% fatality, then and it affects a lot of people, it could. Flu is lower.
Very serious if it really is that high a fatality rate.
The 1918 flu was 2% fatality.
No, understood.
Okay.
Understood.
Yeah.
Understood.
So if millions of people are infected, that's a problem. So
exactly.
Right now, flu looks a lot worse, 15 million, blah, blah, blah. But
by the way, that's all that malaria kills.
The other important thing to remember here, though, is there's actually something you can do about the flu. You can get your flu shot. Right. So we've got something for that. We don't have anything for the new coronavirus that was just discovered a few weeks ago. So don't be all fretting about that and then skipping your flu shot.
That's why if there's a new flu strain, a pandemic strain, and no one's immune, there will be we'll podcast about it and people will listen because it's a new thing. Right.
Well, and you could also say, don't drink and drive. Don't text and drive.
Yes.
Wear your seatbelt. Get your measles shot, get your tetanus shot up to date, a lot of other things.
Get 8 hours of sleep and plenty of exercise.
Then it's, worrying about coronavirus.
Stuff that is going to kill you, yes.
Okay, go ahead.
Sophia continues. Number 3, could you talk more about the flu, please? This year's strain feels like a very nasty one.
We could. I think we did a couple of weeks ago. We had a flu update, right?
Yes. We did.
Sophia, maybe you missed it. Go back and check it out.
Go ahead.
I think if we talked about flu now, people would turn it off. They want to hear about coronavirus. Right?
Exactly.
Yeah, probably true. Thanks for all that you do. Greetings from Greece. And then she continues with more questions. Sorry, you did answer question number 1 on TWiV 582. I listened again and paid attention this time. However, I still have some questions if you don't mind. So how can you shed virus if you are asymptomatic? Like, where does the virus come out of?
It's great.
Which orifice would you like us to talk about?
Anyone want to tackle that?
Respiratory virus is going to come out of your mouth and nose.
Correct.
Whether or not you're sneezing, you're
right.
talking,
Right.
breathing out, eating.
Exactly.
Yeah,
So
all this.
Didn't we do the paper with the guy with the traffic cone on his
Yeah.
head doing the?
Yeah.
it was you. It was you. It
whatever
looked like you.
Symptomatic.
I think that was the
Yeah.
PFU and you're a chew.
Yeah.
That's a great.
Yes.
paper.
But I think Sophia wants to know, how can you be asymptomatic and shed? Well,
Shouldn't you be
That's
sick? Shouldn't you be
that is what is the puzzle. For many viruses, you can actually be have the virus is replicating in you, and you're not sick, but you're shedding. So it's replicating enough to shed, and where you're shedding, it depends on the virus. So, for example, most polio infections, you shed it in the feces, and you're not sick. 99 out of 100, you don't get sick.
Typhoid Mary wasn't sick either.
And if you think about it, if some of the sickness is due to your immune system's response, that you could be shedding the virus before your immune system's response is giving you the symptoms.
Yeah.
Right.
And I think it's generally true that a lot of the symptoms that you experience are the immune response.
Yes.
Yes, they are. The cough tends to persist a couple of weeks after recovery, at which time I am assuming you are not transmitting anything. I had mild symptoms and a dry cough. Still wonder if I were transmitting. How far do these droplets go, and how long can they stay alive on air or surfaces? If you cough on your scarf, can you assume your scarf doesn't contain live virus the next day? If you are lecturing, this gives you some space from students. Can you still transmit? Does the virus stay live and suspended on air just like TB?
Let's do
Depends on the virus.
Let's do the previous one. If you cough, so the peak of virus shedding is usually at the peak of disease, and then it declines rapidly. If you have a cough for weeks and weeks, you're probably not shedding during that time. And there are many studies that
cytokine storms.
cytochrome P450.
Yeah.
That's your immune system lingering.
So droplet science is very interesting. There are respiratory droplets. They're large ones, about 5 microns or bigger. They tend to fall about a meter from you. Okay, and you make those when you talk and sneeze and cough. But so you have to be relatively close to someone to transmit. Like Dickson, I could be transmitting to you now. But the droplet nuclei, which are less than 5 microns, can go for many, many meters. And that's what you can also make those as you breathe. And measles virus is very good in getting into those droplets. That's why it can transmit for long distances. If you cough on your scarf, that scarf will have virus. And depends on what virus, some last longer. Rhinoviruses can last up to 7 days on banknotes as long as there's mucus on it. Mm. So if you blow your nose into a dollar bill, it'll remain infectious for a long time. I don't know how long this coronavirus would last on a scarf.
Well, it's got a membrane, right? And usually viruses that have lipid membranes around them are usually, there are always exceptions, but usually not as tough as viruses that do not have membranes.
I would guess that, so these coronas are transmitted by pretty large droplets, and so they don't stay suspended in the air for long periods. So,
right.
Yeah, if you're lecturing a class and you have some meters between you and them, that's not a problem. However, if you're lecturing a class and you have measles, the whole class will be infected.
Yeah.
Way in the back of the room even. It's amazing.
So if you're feeling sick, you could have flu, you could have a coronavirus, you could have a rhinovirus, you could have an adenovirus, and all of those have different characteristics with respect to these issues of how long they live on a surface and how well they are transmitted through the air.
That's a really good point. The health ministry in Greece is circulating a brochure for schools to follow to cope with the flu epidemic here. It suggests keeping kids away from school even after a day after recovery, still wondering when someone doesn't transmit anymore, and cleaning all school surfaces with chlorine.
Really?
Did you hear there's people who are drinking chlorine?
Oh, man, I don't even want to think about it.
Is it ridiculous? It's always good to err on the safe side. A day after your child has recovered wouldn't hurt.
That's fair. Yeah.
Yeah, I think that makes sense.
But disinfecting surfaces is okay. Chlorine is great for inactivating most viruses, actually. Yeah.
Yeah, so those are very reasonable measures, and I think I got something from my daughter's school. Like one of their standing policies is that the kid is supposed to be afebrile without medication for at least 24 hours before you send him back to school, and that's just the policy.
Yep, that's the same.
So yes, that should they should be over their fever and recovered before you send them back to school, and sure, clean the school surfaces because there's junk on them.
Yep. If you already had a coronavirus infection earlier in the year, okay, you couldn't have known, but hypothetically, I am sure many people have had such an infection without complications. Are you at all protected against the Chinese one? Or is it like the flu, totally new, no protection even if previously infected?
See above.
This is a new virus. You're not going to be protected. There are other coronas that can cause common colds, but this is totally different. So,
right, but I think the question is if somebody had, Sophia's question seems to be if somebody had an asymptomatic infection with this previously, maybe somebody in China.
Oh, they should be immune, yeah.
Are they immune? And yeah, probably.
Probably, yeah.
Hopefully.
Yeah.
And her final question is, how far are you guys concerned, especially if you travel overseas? And how well do you think the epidemic is being handled in China? She sends a link from a trusted French station and was worried about how it's handled.
Yeah, there's a lot of chatter about they're not handling it properly, but it seems to me that they're handling it very well.
According to Tony Fauci, and he answered that question last night too, he said they're answering it slower. There's no real-time data, so that you don't have you have a day or two lag in what's going on in China right now.
I am sure they are pressed to the limit for what, all these diagnoses that they have to do and then isolating patients and so forth.
They don't have the news media outlets that we do.
Is that right?
They're not used to doing that.
They're handling it a lot better than they handled SARS.
Well, you know that.
There's been tremendous progress on that.
That's not saying anything.
Kudos for that. No, I think that's saying something important, that they have learned from that experience and have adjusted things, and that's great. And really, in terms of the handling of the epidemic, the only thing that I would criticize is the lockdown, which is counterproductive.
One of the new graphics on the South China Post explainer is how many people are affected in this lockdown, and then comparison to populations of various places. And right now it's around 60 million, and that's the population of Italy.
Right.
Wow.
So, yeah.
To answer her question directly, I personally am not concerned at all, okay, for my own personal safety, okay? And, of course, I don't have any overseas travel planned, but if I did, I might think twice before flying into Wuhan, not that I have a choice anymore, okay? But even traveling to other areas of China, I don't know if I'd probably look at a map and see how affected it was, but I don't, I wouldn't be all that concerned. I wouldn't be concerned at all about flying somewhere else. As I've advised several people who've asked me the same question, probably the most dangerous thing you're going to do is drive to the airport.
Mm-hmm.
Right.
Mm-hmm. Yeah.
Are you concerned, Dickson?
Nope.
How about you, Kathy?
No.
How about you, Brianne?
Nope.
Alan?
No, not especially. My concern, if I were traveling to China, would be that I might get stuck there.
Yeah.
But in terms of the virus, I'm not particularly worried.
My nephew just returned from Wuhan. He was there.
Is he okay?
Yeah, he's fine, and so am I.
Look, China's a good place to be stuck. They have good food.
They do.
Well, unless the city's been locked down and they can't get supplies, then that could be a problem.
Well, he missed one day of that one. He almost did get stuck.
All of us are saying we're not worried because it's not here in any numbers. But let's say we start having tens of thousands of cases. Would you then be worried for your own health?
I'll have to see the fatality rate.
Exactly. Not based on the case reports of who gets really ill and who dies from this.
Remember, most of the deaths are in older people. But
with
I'm
underlying conditions.
I'm over the cutoff.
I'm way older than you.
Now, let me just say, if someone invited me to Wuhan to do a TWiV, I'd go. Because I think it would be fascinating to be there and talk to these people who are doing this work. I would just be careful.
Wash your hands.
I would wash my hands. I might even wear a face mask, even while recording TWiV.
That's right.
I would remind people we've done, over the last several years, we've done SARS, MERS, Zika, the 2009 flu pandemic, okay? And
Ebola.
And Ebola, that's right.
Yes.
Right? And
Amazing, isn't it?
with the possible exception of the 2009 flu pandemic, where some people probably got sick with it, my guess is that you haven't been seriously inconvenienced by any of those.
Right.
Now, I do pay attention. My prediction is that this will burn out kind of on the same order as SARS did, but I'm watching.
Yeah.
See what happens.
I would go with you to that TWiV in Wuhan.
Yeah.
Good.
We'd all go.
Yeah, can we all go?
Listen, my first trip to China was to interface the Columbia University School of Public Health with the China National Public Health Service to see if we could be of assistance to them in rearranging how they deal with these outbreaks. And it was very instructive because they still didn't know how it was transmitted when we got there. And all of the staff from that meeting from China was fired 3 weeks later.
Yeah, you said that last week.
Yeah. Well, I'll say it again.
When I was in
Tandem
Singapore,
repeat.
there were several scientists from Wuhan, and they asked me if I'd ever been. I said no, and they said, we will have you in Wuhan. So now I'm not saying this lightly. If they email me next week and say, come, I'd go. Now, probably my family would.
As long as you can get a return flight.
They'll stay there for a while. What the heck?
Stay there for
It's a very modern country. I understand, and they're very interesting things to look at.
Lots of interesting
I bet they have
things.
microphones and recorders. I'd bring my own.
Yeah.
We do.
Let's do some picks, folks. What do you think?
All right.
Yeah.
Sounds good.
Brianne, what do you have?
Yes. I have a Netflix series that was just released with rather good timing called Pandemic. And so it is a 6-episode documentary series that actually talks about both public health measures and research into different infectious diseases.
Excellent.
So the first episode is all about the flu epidemics seen in Asia and making a universal flu vaccine. And I've already had a number of students come in to ask me questions after having watched it. The second one is all about vaccines and sort of whether or not to vaccinate against Ebola, as well as sort of the anti-vax movement. And so it hits a lot of the subjects that we've been talking about, and I found it to be pretty interesting. I've been watching through it to try to show some clips in class.
Cool.
Nice.
Yeah.
Who made it?
This is who made it? Well, it's a Netflix original, right?
Yes,
Yeah,
it
but
is.
are there people that are recognizable in the documentary?
This is guy Dickson Despommier.
No. You don't want him. You don't want him.
I don't know.
I have not seen anyone recognizable, but
Okay. Like WHO Phil Schultz or Tony Fauci or something like that.
I have not seen Tony Fauci. I do not recall if there was.
Or the head of CDC? Yeah. Okay.
I do not
All right.
recall all of the people I have seen.
Alan, what do you have for us?
I have a book pick. This book came out in 2018. I think I just got around to reading it. It's called Educated by Tara Westover. It's a memoir. I don't normally read a lot of Westovers. A lot of memoirs. Nothing against Westovers. I sometimes fly into an airport called that. Anyway, this book is really, really good. She is a fantastic writer who has had an extraordinary life. She was raised in a survivalist family in Idaho and didn't go to public school and was not exactly homeschooled either. She just kind of grew up there, and they lived apart from society, and her mom worked as an unlicensed midwife, and her dad ran a junkyard, and it's the story of how she grew out of that and managed to essentially bootstrap her way into an excellent education. She taught herself what she needed to know to pass college entrance exams, got into Brigham Young. After getting a degree there, then went to Cambridge, England, and has since received a PhD in history from there, I think. And it is a really extraordinary story, and I highly recommend it.
Nice.
Cool.
All right.
Yes.
Yeah.
On the list.
And I see my little library app that you recommended
Yes.
last week comes up, and I'm going to have to wait for this because they're.
But you can click it. Yeah, I did too.
I can at least put it in my queue.
I clicked on my local library and I logged in and I put a hold on it.
Yeah.
And about a week later, they said your book is here, and I swung by and picked it up, and that's how I read it.
Nice.
Great.
Rich Condit, what do you have for us?
Relative to the episode, on a lighter note, I'm picking the new series Star Trek Picard.
That's right.
Okay. For you Trekkies out there, and, I think everybody probably ought to. Well, no, it takes a lot, of background. This is on CBS All Access, and so they got me. I subscribed to CBS All Access in order to do this specifically, because that's the only place you can get it. It's a new Star Trek series in the franchise that stars the Patrick Stewart, who plays Jean-Luc Picard, the captain of the Enterprise in Star Trek Next Generation, which was one of their best series, in my opinion, and the opinion of several others. And this season has 10 episodes. They've dropped 2 so far. I've watched both of them. They're terrific. So if you have any interest in this sort of Star Trek stuff, this is good.
Cool.
I have never seen a Picard Star Trek.
Oh, they're great. Oh, they're terrific.
When I was a kid, I used to watch the other ones, but that was it. I never saw any.
Next Generation. Now, I haven't seen all of the series, but Next Generation is really good. I promised myself before I retired that one of the first things I was going to do was binge Next Generation again. And I started about a year or so before I retired and binged it in a couple of weeks.
Wow. Good for you.
That's the Picard of the week.
Dickson, what do you have for us?
Well, I have an interesting new development with regards to a telescope in Hawaii that's been named after the senator from Hawaii, Inouye, Daniel Inouye. It is the highest resolution photograph ever taken of the surface of the sun.
I was going to pick this telescope as my pick this week. And
well, tough darts.
Yeah.
It is
great.
Amazing. And there's video with this as well. And it is just stunning.
This is an earthbound telescope?
Yes, it is.
Yes.
It's on the Maui, I believe it's on.
Okay.
Yep.
It's unbelievable. We have no idea of what goes on the surface of that sun.
This big picture looks, like something I've seen before.
Well,
I mean, I know it's not, but
mildly. No, they've got
macaroni and cheese.
Something like that. Yeah.
Corn.
Corn. Yeah.
No, they've taken lots of pictures of the surface of the sun from another space telescope as well, but this is the highest resolution picture ever.
This is on Earth. So
it's on Earth.
Yeah.
So give me an example of the magnification. What are we talking about here? How many miles across is this picture?
There's an image that shows
as big as Earth.
Well, there's an image that shows
I see it there,
a zoom in of the Earth, and that shows Texas superimposed in a tiny little square of one part of that image.
That's right, exactly.
So
wow,
it's amazing.
Okay.
Wow.
And the resolution of the flame, the solar flare that's just at the little surface, is absolutely perfect.
Stuff is going on there.
Stuff is going on there.
So this is the picture that was on the front page of the New York Times yesterday?
Correct.
Yeah, okay. Correct. And that's where they said kernels of corn or something. Yeah.
Kathy, when you say the front page, do you mean the web version or the paper version?
The paper version, Vincent.
You really you get the paper version? Yeah.
Yes.
Awesome. You get it delivered, or you pick it up at the local?
No, I get it delivered. And you can also get a version online that's, I forget, it's just jumped out of my head. It's called Replica Edition. And you can actually see the ads and everything online. So you're flipping through the pages like on your computer or your tablet or whatever. And so when I travel, I can get it that way if I want.
We just get the Sunday version. Yeah. For the puzzles and stuff.
I was some hotel I was at recently. In the morning they had a copy of the Time, and it was like the first time I'd seen a paper copy in a long time. Yeah. It was really cool to pick it up, open the
Tell me about
pages.
it.
That's a good experience because we grew up with it.
I agree.
Yeah.
I agree.
I get to do it every day.
That's wonderful.
What do? Do you read it in the morning, or do you take it with you?
You know,
No,
you
I
can pay
don't-
somebody to deliver it to you every day, Vincent.
Well, I used to actually. But go ahead. Kat,
I typically look at the front page in the morning, and then as much as I can of it in the evening, and then there's this stack of papers, and then every now and then I just have to purge them all because I didn't get to them. But I try to get through the front page section.
I had a friend who got a subscription to the Times, and he said, the first thing I do is I check the obituaries to make sure I'm not in it, and then I read the rest.
Yeah.
Well, when I lived in Manhattan, I used to pick it up on the corner before getting on the subway, and then
right.
moved to New Jersey. I had it delivered, and I would get it at the end of the driveway before I left for the train, but often it wouldn't come in time, so I got pissed off, and I just restricted it to the weekend.
Right.
And then the delivery was so erratic, I just canceled the whole thing.
Too bad.
Now I pay for online only, but that's kind of a nice.
But we get the Sunday.
You may be able to get the replica edition if you're already paying for online.
Right.
And I miss using the newspaper to start fires.
Yeah.
I don't have any fires.
Or to
Don't
clean
you
the
get
windows.
those flyers in the mail?
Yeah, that's what we use.
Ad
Yeah.
flyers.
You,
yeah.
you can clean the windows of your car like that,
But
too.
the Times was so good to crumple up. That was the best.
This is
A lot of ink. A lot
This
of
is
ink
why
would
people
come
call us chatty, because this was all
yeah,
about
it's
I'm
exactly
sorry.
the same thing.
It's like it's like a plaque assay. Should we switch to plaque assays now?
Kathy, what's your pick?
Well, like Alan, I have a long list of things queued up for picks of the week, and some of them are from the University of Michigan, so I have no danger of Alan picking them first. This one I picked because it's about vampire bats and trying to vaccinate them. And so one of the diseases that bats get is rabies. And so they're talking here about a vaccine for rabies that can be applied orally or topically and then be spread between bats. And those have already been shown to protect multiple bats against rabies in the laboratory, but they had no way of ascertaining how good the spread is in the wild. And so they used a glowing fluorescent gel and put that into the vaccine.
Oh, great.
And figured out, this is published in Nature, Ecology, and Evolution, that so it's rhodamine B, and they tried it in vampire bats in 3 places in Peru. And when the bats groomed each other, it led to fluorescence in their hair follicles, and they could monitor that by fluorescence microscopes and show that orotopical rabies vaccines would protect 2.6 bats for every bat that's vaccinated. So it's kind of like the opposite of an R naught, you know?
Yes.
That's RNOT.
Right. So anyway, I thought it was pretty cool. Yeah.
That is cool.
Yes, rabies is the one virus that makes bats sick. That's why they're erratic. If you see a bat coming near people or a raccoon, they're erratic.
So
from the
if
virus.
you gave a rabid bat the vaccine, would it cure them?
How are
Rabies
you going to
vaccine?
give a rabid vat?
You give the
Are you
bat
gonna touch
rabies.
it?
You give rabies
Depends.
to the bat to begin with.
Depends how long before they were infected. With people, you
Yeah.
have 2 weeks.
Yeah,
right?
exactly.
Yeah.
Right.
I don't know.
With people, once you have symptoms, you're dead.
No, that's true.
Right.
Except for 2 people.
And they would rather be dead, right?
They would rather.
Actually, one young girl who they put into one of these comas, she went to college. She's
Yeah.
okay. Yeah, we covered that a long time ago on TWiV. Yes. My pick are my virology lectures for 2020.
Nice.
Mm-hmm.
They have started. We're on the 3rd lecture, so all the lectures get posted on YouTube, free, and you can go check them out. They're a little different this year. I'm adding in every lecture a little reference to the new corona when it's relevant, and there's some other things that are new too. It's my 10th year of teaching this undergraduate virology course,
Nice.
and
How many students,
it's
Vincent?
106 students. It's a
Wow.
good.
It's a
Someone,
big load.
professor down there said, you have one of the biggest enrollments in the spring semester. Can you imagine on Columbia University campus?
Wow.
Cool.
Right, people want to learn virology. So I thought
Great.
this 10th year would be a good year to stop, right?
Why would you stop? Don't you like it?
Because I don't want to stop on the 11th year. That would be horrid.
Don't you like to do this?
Oh, I
don't know. 11's a prime number. That's good.
Why would
No, but then a student came in after the first lecture, said, I can't take it this year. Will you teach it next year? And I said, if I'm alive, I will teach it. What a morbid thing to say, right?
You'll be alive. You'll be alive.
We have 2 listener picks. One from Edward. Hello, the TWiVerers. That's cool.
TWiVerers.
The TWiVerers. Quick listener pick, and this is old, I'm sorry. If TWiV listeners fancy adding some virology to their festive decorations, then they might like to check out this booklet of paper snowflake designs. Each one is a reasonably accurate virus capsid. The attached PDF can be downloaded from the MRC University of Glasgow Center for Virus Research. And there's a link. Thanks for the show. Ed is a research fellow at the Glasgow Center for Virus Research, which I visited. Lovely place. Glasgow is a lovely town.
It is.
I really like it. And from Anthony, microbe hunting from autism to Zika. Ian Lipkin, a YouTube video, possible listener pick. Sure. So I think it's a bit old.
2016?
Because he looks younger. It's a keynote lecture that he gave somewhere. Good time to talk about virus hunting. Thank you both for your picks. That'll do it for TWiV 585. Microbe.tv/TWiV for the links. A lot of links these last few weeks. A lot of papers we put in there. If you want to check them out, you can go there. If you listen on a phone or tablet, on a podcast app, please subscribe so that you get every episode for free. And we know how many people are listening. And send your questions and comments to TWiV@microbe.tv. We have a few more corona questions we'll get to next week. If you have more, send them in. And consider supporting us if you like what we do. Microbe.tv/contribute. Dickson Despommier, verticalfarm.com. Boy, that's an old one. And parasiteswithoutborders.com. Thank you, Dickson.
Welcome, Vincent.
Have a good course today.
Had a wonderful course today.
What did you talk about?
Talked about the Gaia hypothesis and evolution according to Darwin.
Who else is it according to? Or would be? Thank you,
We have
Dick.
many choices on that one.
Thank you, Dickson.
You're welcome.
Brianne Barker is over at Drew University on Twitter. It is BioProf Barker. Thank you, Brianne.
Thank you. It was great to be here.
Good to have you back. Kathy Spindler, University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor at the University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
You know, Dickson was in Gainesville not too long ago. Did you know that?
I told him that.
Did you tell him that?
Didn't I tell about last week?
Meeting its president. Yep.
You forget, I forget.
Last week.
Okay.
Alan Dove is at turbidplaque.com. On Twitter, he's Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV, and Ronald Jenkins for his music. This episode was recorded, edited, posted, produced by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.